from understanding nothing and almost every patient dying miserably, to giant understanding and bringing almost everyone back to health. The story of the Philadelphia Chromosome and the 20th century. The sequence of scientific discoveries that this video will trace is beautifully summarized in Jessica Wapner's book. The way the Philadelphia chromosome led to CML was a story that unfolded like 100 painters applying brushes to a canvas at some time or other over 25 years, driven only by curiosity and sometimes a vague hope that their work might eventually be relevant to human cancer. There had been no final picture in mind and no awareness that they were even painting something together. And yet there it was, a scientific masterpiece. So go get the book and read the book. The illness we're talking about is chronic myeloid leukemia, CML. This is a remarkable story, one of my very favorite stories in science. Dozens of scientists who, working over decades, had barely an inkling how their little bit would contribute to a pretty phenomenal final achievement. Ably assisted by chickens, mice, and leukemia patients. From an improbable start, through seemingly separate threads of scientific unfoldings, peppered with good luck, bad luck, mistakes, innovation, controversy, curiosity, and incredible persistence. Patients with chronic myeloid leukemia, CML, have too many cells from the myeloid or myelogenous branch of the immune system. Here's a sample from the blood of a CML patient. You can see the excess of the abnormal white blood cells. There's too many of them, staining purple. Those purple cells, they crowd out normal cells in the bone marrow, which is where, after all, these myeloid cells develop. It was chronic, meaning that it could take several years for the lethal effects to show up. But eventually it would go into blast phase, where these cells, called blasts, would start multiplying really quickly. Crowding out red blood cells, the patient feels profound fatigue. Crowding out platelet production, the patient cannot clot and starts bleeding everywhere. Crowding out white blood cells, the patient suffers uncontrolled infections. So it's bad. And it was almost always fatal. If you're the hematologist taking care of the patient, it's tough to watch. You have strong motivation to find good treatment for these patients. But Dr. Brian Drucker and others used decades of science to find a great new treatment for these patients, a new class of wonderful medicines. It took dogged determination through tough times. I will put a link to a talk Drucker gave. It's personal and patient-oriented. The story of the science starts in 1959 with this terrific discovery called the Philadelphia Chromosome. Like many important scientific discoveries, it was an accident. 1959, only 6 years after the identification of the double helix structure of DNA, and only 4 years after the first accurate count of human chromosomes by Jo Hin-chio. So here is a chromosomal spread, 46 chromosomes, but in pairs, so 23 times 2. These chromosomes are numbered according to their size. Number 1 is the largest. Peter Noel and David Hungerford decided to take a look at a leukemia cell from a CML patient. Noel took a shortcut and just used tap water to wash the sample, and lo, this is what he saw. The leukemia cell's chromosomal spread. There's something very peculiar right here. Something tinier than the tiniest chromosome. And this tiny thing would show up in patient after patient with CML. Terrific, but what does it all mean? This curiosity was ignored at the time. Many years passed, 13 or 14 years, and there was a new technique. It was possible to look at those chromosomes and know which one was which. You could see these patterns of light bands and dark bands that were characteristic of each individual chromosome. You didn't have to just guess at how big each one was relative to the others anymore. Janet Rowley had mastered this technique and applied them to CML cells. She saw something provocative. On chromosome 9, one of the copies has this little extra bit here. That process is called translocation. But that's not all. Look over here. Something broke off of one of the copies of chromosome 22 and stuck onto one of the copies of chromosome 9. What remained was the Philadelphia chromosome. How did she know that? Because the amount of extra material in chromosome 9 exactly equaled the amount of material lost from chromosome 22. 14 years prior, Noel and Hungerford, who discovered the Philadelphia chromosome, did not know anything about the involvement of chromosome 9 and chromosome 22, so that's a big new clue. Spoiler alert, haha, irony alert, looking at that big new lump under chromosome 9. We would guess that that's causing the leukemia. But no, even though chromosome 22 got shorter, it's extra new material from 9 that went to 22 that's causing the leukemia. But it was far too small to see, not something that Dr. Rowley or anybody else could have guessed at the time. But what's going on here, and what does that have to do with patients being sick and dying? Wind back the clock a half century. Something was going on way back in 1909. There was a chicken. There was a chicken with cancer, a kind of cancer called sarcoma. A farmer walks into an institute with a chicken. She wants Dr. Rouse to remove a tumor from her chicken. He cuts out the tumor. So here is Rouse's chicken. Well, really, the lady's chicken. But the chicken died, so that's the end of the story, right? Not the end of the story. The audacity of that woman bringing her chicken to the Rockefeller Institute launched an incredible series of scientific discoveries, but only after another half-century had passed. Dr. Rouse couldn't have guessed that, but he just got curious and discovered that, like a lot of infectious diseases, this sarcoma could be transfected from one chicken to another in the same way that an infection with a bacterium would. But strangely, it was not a bacterium. By straining through a material so fine that only a virus would be small enough to get through. Rouse had shown that it had to be a virus. Eventually, Rouse's sarcoma virus, later called RSV, was discovered to be the cause. Moving that RSV virus from one chicken to another made the second chicken get that sarcoma. For quite a while, people were speculating, is cancer in general caused by viruses? Could human cancers be caused by viruses? For human CML, this is the wrong species and the wrong cancer, and later we'll see it's the wrong gene. And CML is not even caused by any virus, never mind this one. But this chicken begins a thread of scientific discovery and a thread of irony. We'll see scientists pursuing viruses that cause cancer in chicken and mice as we trace through those threads through the 20th century. The critical insights will converge on understanding and curing a human cancer not caused by a virus. That's science for you. That cancer virus idea almost died out, since the common cancers in people do not spread at all like the known viral diseases. But stick around for the virus wrap-up at the end. Fast forward, way forward, a half century after Rouse's farmer's chicken. Let's see what the mice have to say about all this. In 1969, 10 years after the discovery of the Philadelphia chromosome, Herbert Abelson was studying a leukemia based on a different kind of immune cell, a T-cell leukemia virus. He took out the target organ, the thymus, just to see where else the presumed virus might infect. In Jessica Wapner's words, the reason for the experiment was simple. He was curious. Like many experiments done with no idea that they would be turning points, just for curiosity. Abelson accidentally found a new virus that was causing a B-cell leukemia, yet another kind of immune cell. Now that virus is called Abelson murine leukemia virus. Murine meaning mouse, AMLV for short. Cancer viruses for mice and chickens? How about people? Hold that thought because we're going back to chickens now. Hey, remember that sarcoma virus that Dr. Rouse had discovered? Now called Rouse sarcoma virus RSV. In 1970, Stephen Martin wanted to find the part of RSV causing the cancer. He created a set of temperature-sensitive mutants of RSV where he could turn on or off a gene just by changing the temperature and see if it was needed for the cancer. He did it. He found the cancer-causing bit. Since it had caused that chicken sarcoma, he called it SARC, SRC, as in sarcoma. See you later, chickens. Hello again, mice. It was now 1975. What nobody had done yet was to take normal animal cells and watch them in vitro in the petri dish as a cancer gene made them cancerous, to uncover the mystery of how it was happening. That's what Naomi Rosenberg wanted to do. Working with Chuck Shearer, she looked around and saw that Abelson's virus might do that in a mouse tissue culture. How do you find mouse cells that you can culture? Finally, they found that liver cells from mouse fetuses would work. I tip my hat to all the scientists. The grueling, slogging part, trudging through many failed tries. That technology for transforming cells with a virus bridged critical breakthroughs to come. Next, Rosenberg was working with Owen Witt, who brought expertise in antibodies for viral proteins. He picked a viral protein called GAG to start with. It was one choice out of several. His antibody to the GAG protein detected a surprise, a funny new protein, an extra large GAG variant, and that was causing the cancer transformation. So even though GAG itself was not responsible for the cancer, starting with GAG was a lucky choice. Whatever the extra part was, it was presumably some gene that was responsible for the cancer transformation. And since it was from the Abelson virus, they called it ABL or ABL. This combination of a common viral gene and a cancer gene would be called GAG-ABL. Meanwhile, back at the chicken coop, a couple of years go by. Michael Bishop and Harold Varmus studying the chicken cancer. At the end of 4 long years inventing and struggling with a clever experimental protocol, developed a probe with just the bit of virus RNA causing the chicken cancer. They slapped their probe against the chicken genome, and guess what they found? The gene causing the cancer has its match in the chicken genome itself. They showed that normal genes in a chicken have the potential to become cancer genes, oncogenes, sitting quietly in the chicken, mouse, or person, behaving themselves, doing their appointed jobs inside the normal, healthy cells. They're called proto-oncogenes, but they won't become actual oncogenes until what? If the cancer-causing gene is already in all of our cells all the time, Why don't we get cancer all the time? There are several answers to that, but we're going to find one powerful answer related to a cancer of people, CML. Next year, 1977, Erickson and Collett discovered the protein product of the Src gene that Stephen Martin had discovered a few years earlier. And what was it? What protein was that gene producing in the cells? It was a kinase. Now we're getting somewhere. A kinase, an enzyme, a biochemical catalyst. That means a molecule that makes a difficult chemical reaction easy. A kinase catalyzes the transfer of phosphate groups from the high-energy ATP molecule to specific receiving molecules called substrates. This process is called phosphorylation. The substrate gains a phosphate group. The ATP molecule donates one of its 3 phosphate groups to the kinase, which then passes it on to the substrate. Phosphorylation turns out to be shot through all kinds of processes in all of the cells of the body. So the Src gene makes a kinase. That's fine. That's just great. But it's a chicken cancer. So what? A couple of years later, Tony Hunter was working on the polyomavirus. He discovered it had kinase activity. He was trying to find out what amino acid was being phosphorylated by the kinase. Surely it was threonine or serine, the usual suspects. But one day he didn't feel like making a new buffer for the experiment. The reused buffer revealed something unexpected. The amino acid was tyrosine. People hardly ever thought about tyrosine at the time. Immediately, Erickson had to find out if the target amino acid for the Src chicken virus kinase was also tyrosine, and it was. This was 1980. The midpoint between the original discovery of the Philadelphia Chromosome in 1959 and the final approval of a miraculous kinase inhibitor drug for CML patients in 2001. And also in 1980, Witt and colleagues found that the Abelson-Murine leukemia virus protein is itself phosphorylated. Sometimes kinases actually phosphorylate themselves. Tyrosine, chicken virus, mouse virus. Who cares, right? Just some picky minutia, right? How about leukemia in people? By now, all the patients benefiting from kinase inhibitor treatments care a lot. We'll see how this tyrosine thread weaves among our beloved chickens, mice, and people. Back to the people world, then. Owen Witt was now studying the chemical properties of CML cells and found that they have a very active kinase, but not a normal version. It was fused to another protein, a mystery protein. You dig it? In the mice, a fused mystery protein. In the human patients, a fused mystery protein that reacts to the same antibody. Sounds like Agatha Christie fuses with Hercule Poirot. In the early 1980s, the discoveries about human CML are coming fast and furious now. I'll connect names to discoveries, but honestly, all these people were finally working together now. Naming every collaborator on every discovery would get tedious, so forgive me when names are omitted. Imagine the phrase, "and others," sprinkled everywhere. Owen Witt's team learns that the kinase in CML is always active, always switched on. I wonder what that kinase is and what that kinase does. Groffen and colleagues made a probe to detect the Abel gene. The GAG gene belongs exclusively to viruses, but Abel was a mouse gene. Could Abel also be a human gene? Yes, Heisterkamp and Groffen's team shows that Abel is found on the normal human chromosome 9. Whoa! And only on the normal chromosome 9, not on the chromosome 9 that swapped pieces with chromosome 22, even though it's bigger. Whoa, ho, ho! Did that remind anybody of the translocation that made the Philadelphia chromosome? You bet it did. Annalise Decline found that the Able gene, which should be on chromosome 9, is now on the Philadelphia chromosome, squatting on the old chromosome 22, and not found on the chromosome 9 piece that had swapped with it. And how about the other part of the Philadelphia chromosome? Well, Graffin's gang find a gene where the breakpoints are happening, calls it the, quote, breakpoint cluster region, or BCR. It is on the normal chromosome 22, and now we know that the Philadelphia chromosome has a fusion of BCR with Able. That is Witt's mysterious fused kinase. Later on, Eli Kanani found that BCR-Able made a much longer message RNA than the standard issue Able and was produced in great quantities. So like GAG-Able, BCR-Able made a bigger protein than just Able. In 1984, Owen Witt discovers that cells from a CML patient contain, sure enough, a tyrosine kinase that puts a phosphate group onto the amino acid tyrosine. That kinase serves as an on-off switch for cellular processes. In this case, the switch was stuck in the on position. And what did that kinase turn out to be? ABL, Able, the same protein that is coded by the proto-oncogene stolen from a mouse, or maybe an ancestor of the mouse, by the Abelson murine leukemia virus, maybe thousands or millions of years before Dr. Abelson came along. Now we think we can see what's going on. In the normal course of life, some wayward myeloid cell undergoes a swap between the end of a chromosome 9 and the end of a chromosome 22. This takes the Able gene into unfamiliar territory, glued to the BCR gene from chromosome 22. Maybe the careful regulation that Able enjoyed back home on chromosome 9 is gone. Remember, in the Abelson mouse virus, the Able gene combines with the GAG gene to make GAG-Able, which will cause leukemia in mice. Could it be that this juxtaposition of these two genes that don't belong together could be causing CML? That's very provocative. But not yet proof that this is causing the leukemia. Before answering that, just for giggles, a technique called spectral karyotyping colors each individual chromosome by a special combination of fluorescence. You get these beautiful pictures. This was not instrumental in the development of the treatment for CML that we're creeping up on, but still, you gotta see the picture. And there it is again on the book cover. But could we find convincing evidence to prove that this BCR-Able fusion was in fact causing CML, causing leukemia? Well, in 1990, years later, came the final proof. George Daly, yet another member of the Baltimore lab, had finally accomplished this remaining feat. Daly took one group of mice and filled their marrow with the mutant BCR-Able gene present in the Philadelphia chromosome. Next, he destroyed the bone marrow of a second group of mice with radiation. That would kill the mice unless you put back bone marrow stem cells to grow more cells. He injected the second group of mice with the marrow from the first group. Now, if that was normal marrow, then the second group would do okay. But they weren't normal. The second group of mice developed CML, and that experiment established that the mutant chromosome, and therefore its protein product, BCR-Able, is the sufficient cause of CML. Okay, leukemia in mice, but still the BCR-Able came from people. It's as close as you can get in an ethical experiment. But again, what to do to help patients? Ordinarily, the able is carefully regulated in cells, turned on and then when no longer needed according to the current demands to the immune system turned off. But with mutant BCR able, that process never turns off. It's uncontrolled. It starts spewing out the able tyrosine kinase messenger RNA nonstop. So now the cell is flooded with kinase protein. This haywire activity causes the body to produce too many white blood cells. That is a hallmark of CML. That's the disease. But if you block it with some kind of a drug, then all of a sudden, this kinase cannot get the phosphate and transfer it. The cell division of the myeloid leukemia cells is now under control. The number of cells plummets. So that's the hope, but what are you going to do? What do is you hope there's a guy lying around on planet Earth whose name is Brian Druker, and indeed, here he was. It was 1990, just 6 years after people finally knew for sure what the Philadelphia Chromosome really was, and the same year that Daly finally proved for sure that this bizarro human gene chimera, BCR-ABL, could cause leukemia, at least in mice. Pretty strong motivation for searching for an inhibitor to turn into a medicine. Years later, Drucker said, I was 5 years in the lab and I didn't have a lot to show for it. By 1990, just when I was getting married and settled down in a house in the Boston suburbs, I was starting to wonder what I was doing with my life. Why don't I work on a human disease caused by a kinase? Following a series of crises, losses, disappointments, and broken promises, his career was in a shambles. But he was able to screen compounds from the company Ciba-Geigy to find one that would block ABL and kill CML cells. He tested many molecules on many cell lines, and some of those were control cell lines, not CML samples. He was blinded to which is which to keep any potential bias out. When he did find a promising one, his result was confirmed in a different lab. That's the way science at its best operates. After lots and lots of work, he was able to kill cell cultures of CML with a substance which Nick Leiden, Alex Matter, and others at Ciba-Geigy had developed. But the paper reporting his result was rejected by Science and by Nature, the two top journals for top scientific discoveries. There was another big obstacle to moving forward. CML is a relatively rare disease, with just a few thousand new patients each year. That sounds like a lot to me, but to a drug company, in the eyes of the great engines of commerce, those thousands are nobodies. If there's a small market, there's little motivation for developing it further. Ciba-Geigy deserves credit for allowing this research to continue, pursuing a possible new drug that seemed to have little prospect of profit. Remarkably, years later, it turned out a lot better financially for the company than expected. But long before that, disaster struck. A lot changed when Ciba-Geigy competitor Sandoz made a deal with Harvard. The exclusivity meant Dr. Drucker could no longer work with Ciba-Geigy unless he left Harvard, which he did. He settled at Oregon Health Sciences University and continued to work with Ciba-Geigy, where his collaborator Nick Leiden designed and created what appeared to be an inhibitor for BCR-ABL, a tyrosine kinase inhibitor specific for that bad actor on the Philadelphia chromosome, testing the new kinase inhibitor on bone marrow samples from patients. They got the final confirmation it worked on human CML cells. Again, a shout out to the patients that allowed scientists to take samples so that the research could
advance towards good medicine
using a technique called fish.
By the way,
fish swimming in the ocean.
There's no such thing.
That's a good story. Good book.
By Lulu Miller.
But there is fish.
FISH.
Fluorescence in situ hybridization, where you can take antibodies that are able to detect the ABL gene and the BCR gene and color them with different fluorescences, red for ABL and green for BCR. And if you add those into the samples, anywhere that you would see red and green together, which make yellow, you would be seeing Philadelphia chromosomes. So let's see, where do we see that? Those are yellow right there.
So finally, with this evidence, the company Ciba-Geigy said, okay, I guess we'll try it. We have to test it in people. Entire human patients, not just their samples. That's a giant challenge.
It's one thing to have a molecule. It's quite another to have something that you can actually
turn into a medicine that the patient can actually receive. You have to formulate the molecule into a deliverable, bioavailable drug. Most anti-cancer drugs are given with injections or infusions. That was the plan. But first, before trying a new drug on any patients, there need to be toxicology studies in 2 animal species before launching a clinical trial with the first humans. The company would then continue with a trial using daily injections. Being able to just take a pill would be far nicer. But the word was that it couldn't be made into a pill without destroying the activity. In the meantime, bad luck. That turned into good luck. A toxicology study of the injection in dogs produced severe and deadly side effects. That led to a halt in the plan for the clinical study in patients. But meanwhile, at Ciba-Geigy, Alex Matter pressed on, fishing for a pill formulation, and he got what he wanted, with activity against the ABL kinase preserved.
Luckily, they found that it could be made bioavailable orally. That is, you could take a pill. You wouldn't need a shot in the arm
or IV
to get it in there.
In one of those great ironies of this story, without the delay from the dog toxicity study, the first trial with people would have required injections, but now it would be a pill, so much easier. The research name for this pill was STI-571, but it would soon be called imatinib. The suffix nib indicates a small molecule inhibitor of a kinase. The name imatinib would make more sense, and tinib, the T is for tyrosine. So it inhibits, specifically, a tyrosine kinase. Peeking ahead a couple of years, eventually it would be known as Gleevec. Peeking ahead a couple of decades, over 50 of these tyrosine kinase inhibitor medicines have been approved by the FDA. Imatinib was the very first one. Clinical testing with people began in 1998.
They made the plan, they got approval, and they executed the phase 1 trial. The very first people that ever get a drug are on a phase 1 trial. You start with a really low dose, and then
subsequent patients get higher and higher doses, carefully monitoring
bed toxicities. You seek the highest dose that seems reasonably safe. The dose
for future studies
is called the maximum tolerated dose.
Check out episode 3 for more information on phase 1 clinical trials.
Finally,
Drucker's team reported the results from the first trial, the phase 1 clinical trial.
This is in 2001 in what is probably the top medical journal in the world, the New England Journal of Medicine. And here is the article, Efficacy and Safety of a Specific Inhibitor of the BCR-ABL tyrosine kinase in chronic myeloid leukemia. And here's the abstract for you.
One by one, they planned to try 14 different dose tiers, starting at the lowest, going up to as much as 40 times the lowest.
Now, what did they find? This is good. Adverse effects of STI 571 were minimal. A maximal tolerated dose was not identified. You know why? Because once they got to 300 milligrams or more, they were able to observe complete responses in 53 out of 54 patients. And eventually 7 of these had complete cytogenic remissions.
At these highest doses where amazing responses were seen, there was still no dose-limiting toxicity. So there was no point in continuing the trial at higher doses. What a remarkable miracle that was. You can smash this illness
With this one drug.
No phase 1 trial in cancer had ever produced results as spectacular as this.
It's really awesome. This is about the time that the human genome
sequence was being completed. It came too late to contribute to this story, but
still
a landmark
worth noting. The next phase is the phase 2 clinical trial, where you take the dose that looked good. In phase 1, many more patients.
Here it is. Over 500 patients at 400 milligrams, and the results were spectacular. 89% had not gone on to the blast phase even after 18 months. Complete hematologic responses in 95%, plus very low toxicity. But would patients beyond that early chronic phase, the accelerated or blast phases, also get help from imatinib? Quickly, an expanded phase 1 trial answered strongly yes. And just a year later, 2002, a phase 2 trial with Moshe Talpaz and 21 collaborators confirmed the good news. 3/4 were still alive a year later. 3/4 of patients, that is, I hope 100% of collaborators. And the higher imatinib dose, 600 milligrams, did better than the lower dose, 400. The stories of the patients helped by all this, given the freedom to continue their lives, which, without this new medicine, were doomed to be cut short. Those patients' stories are amazing. Let's do a little wrap-up of the cancer virus angle. Remember the farmer's chicken in 1909 with a sarcoma caused by Dr. Rouse's virus? 6 decades later, thanks to David Baltimore and colleagues, who initially were laughed at, we learned that in addition to the hard-earned knowledge that DNA routinely copies its information into messenger RNA, some RNA viruses can copy their information backwards back into DNA, retroviruses. Well, CML was not caused by a virus. Most human cancers are not, though we know a few that are, including Burkitt's lymphoma, cervical cancer, some liver cancers, Kaposi sarcoma. But studying cancer viruses gave some crucial clues to stupendous advances against CML and other cancers that kinase inhibitors can attack.